So I'm going to go back into the R code that we've been playing with Biomart, and I'll make a couple of comments about using Biomart. So last time we had some issues getting it to work on this computer, and I know there were a couple of messages on the discussion board about difficulties on personal computers accessing Biomart. We're not going to use Biomart all semester long, so we just really need to get it to work for us for a week or two. So hopefully we can get it going. I think it's working on this computer. The one thing that I wanted to comment on for you guys was here. So when we library Biomart, I think that is fine for us right now. So hopefully you are able to install Biomart and then library it. If you're having trouble with that, let me know. Down below, we're going to use Biomart to do something. We are going to use Biomart here for the first time to access the gene documentation for humans from a particular database. Different databases contain different information. We're going to use this ensemble database and get access to sequence data for humans. Okay, so this is what I tried to run last time, and this works on my computer. Chances are it works on your computer. Try this first. That just establishes a connection with Biomart, specifically the data set within Biomart for humans. And apparently on this computer we have to do this. So if you have trouble, see this as an option. But this has been run, and I'm going to pick up with what follows. All right. So we are going to get the gene symbols. So those are the HGNC symbols, like BRCA1 is a code or a name for a particular gene. Ensemble, a different database, has its own ID for genes. And let's go and extract all of the gene symbols, HGNC symbols, and entree IDs for human genes, known coding regions of the human genome. Okay, we have a connection established with Biomart. We named it that object Mart. So some of the, as we move along through the class, if you create an object in R, like X is going to be an object that is just a single number, it's an integer, X. I can print it, I can do stuff with it. Other objects, there is now an object called Mart in our working directory. So if I say ls, that's like on Unix or Linux, print the objects that are currently defined. There's two objects in our R session right now. Let's get rid of X. I don't want that anymore. So RMX gets rid of X. So the only thing in there is Mart. And if you try to look at it, let's say Mart, sometimes whenever you're using somebody else's software, like through a package, and also when you're doing certain things in just base R, if you create an object, different objects have different types. That X variable that we created was an object, a simple one, just an integer value. And if you type the name of the object and hit enter, or if you say print object name, R will do what it knows to do to show it to you. And different types or classes of objects will look different. Different things will happen whenever you say, for example, print, depending on the class of the object. This Mart object is a special object. It's not just an integer. It's not just a character string. It's not a matrix. It's kind of specialized. And so when you try to look at it, you just get this. It's an object of class Mart, and it gives you a little detail about it. But you don't, it's hard to explore it by just typing it. So just keep that in mind. In a case like this, where this has a specialized representation, there's going to be functions that come with the Biomart package that are really your access point to exploring it. So if you try to print something in R and you don't get what you were expecting, chances are there's more specialized ways built in for you to explore it. So go look at like the help package or the help manual for the package, and there should be instructions in there for working with that object. So just an FYI. By the way, if you want to know what the class of an object is, so X is the number 1, Y is the character string, hello, Z is true false Boolean variable. If I say class, variable name, it tells me these things. So X is to R, it's a number. Y to R is a character, and Z to R is a logical. If I say class Mart, it has its own class. So that Biomart package has defined its own tools. All right, so let's get the data that we wanted. I want the HGNC symbol and the ensemble gene ID for everything in that human genome data set. One way to look at the Mart object, when we printed it, it didn't really tell us a whole lot. There is one function that I'm going to use here that came with the Biomart package. It's a custom function that they built, list attributes. So for this particular connection to the human genome data set, if I say list attributes, it tells me all the things that are in there that I could get as data. And there's lots of them. In fact, I said just give me the first 100. So if I do square brackets in R, like just again as an aside, let's make a little matrix here. I'm going to say X is a matrix. Let's make it the numbers 1 through 9, and we'll make it a 3 by 3 matrix. Looks like that. So if I type a matrix and hit enter, it prints it like you would expect. If I want to pull out a small portion or a subset of that thing, then I can use square brackets. So show me the first row, second column is 4. Show me the first column. I don't specify which rows I want. I leave it blank, and that means show me all of them. But only show me the first column. So it's the first numbers in the first column. If I wanted the first row and the third row and the second and the third columns, I could do it like that. So square brackets enable you to subset arrays or vectors. So if I said X instead was the numbers 1 to 10, and I just wanted the 4th through the 7th elements, I can do that. And one last little tip, if I wanted to do X, but I wanted to exclude certain elements, like let's get rid of the first one and the last one, I can use the minus sign and tell it which indices I want gone. Like that. So we'll get practice with doing that kind of thing, but that's basic like data manipulation in R. That R cookbook that I referred you to at the beginning of the semester is a good place to look for these types of commands. So coming back here, we've created our connection, and now let's actually do a query. Get BM is the main workhorse for Biomart, and once you have a connection made, allows you to actually pull things off of their database. So let's run this, and then to look at it, let's run it first. I think it takes a second. Some of these function calls take a little while because they are connecting through the network to a big database and doing probably a SQL search on the server side. So now we have something called my results, and if I print that, I get way too much output. So that was not the preferred way to look at the output, because if I keep scrolling, I just filled up the whole display. And in fact, it stopped because it reached the maximum number of lines to print. So instead, I'm going to use this, which I mentioned last time, by saying head. Often R knows what something sensible to do when you say that. If it's a matrix, if X is a matrix like we had before, let's make another matrix just real quick. Just takes a second. Let's do 1,000 random normal numbers, and let's make it 100 rows, and it will figure out that it needs to be 10 columns. So I just made a matrix that's 100 rows by 10 columns. If I said X, enter, or if I said print X, it would fill up the screen and have to scroll up to see all 100 rows. If I say head X, it just shows the first handful of rows. That's a handy way to just take a peek at a big object. And just to illustrate using those brackets, I could do that manually by saying, just show me the first 6 rows. Same thing. Over here, I'm going to use the head function, and R has a special method built in for dealing with objects of this class. Head my results, this is what came out. These are the first 6 entries out of however many tens of thousands that we actually have in this object. The first gene in the database has HGNC symbol MT-TF, and that corresponds to an ensemble gene ID of that. If I went to the HGNC website and typed that in, I would learn about that gene. And if I went to the ensemble website and typed that in, I should be seeing it's the same gene that's being described. So now what can we do? Now we could go get an actual sequence for a gene that we're interested in. For example, we'll look at this gene. It's called the Mike gene. And just briefly, what do we know about the Mike gene? Just go into Wikipedia. This gene is important enough that it has its own Wikipedia page. So it is a gene. It's referred to as a regulator gene. And when you see regulator or regulation, think about what we discussed last time about many genes. What they do is they get copied and turned into a protein. And the protein does things physically. Other genes don't necessarily code for a protein, but they might code for other intermediary products that regulate, and by regulate we mean turn on or turn off, turn up or turn down the activity level of other genes. This gene is what's referred to as, it's associated with, it codes for a specific type of protein called a transcription factor. We don't have to know everything about those, but we will use that. We will refer to them a few times throughout the semester, so I'll just mention what they are roughly right now. A transcription factor is a protein that will come to the DNA. So remember, the DNA is just this double helical structure with with bonds in between A's, C's, T's, and G's. So A is connected to T and C is connected to G, going down this ladder, and then it twists into a helix. Somewhere in that ladder, that helix, double helix, is the sequence for the Mike gene, and or for any old gene, let's say BRCA1. In order to generate a BRCA1 protein, to actually put that function into action, like we talked about already, a copy has to be made of the reference manual, the A's, C's, T's, and G's, and that copy goes and is turned into the protein. So just imagine what has to happen inside your cells, however many thousands and millions of times throughout your lifetime. Your body's doing stuff. Proteins are being produced and going and doing their things, and then things are being produced and manufactured all the time. So in order to do that, you have to come to this double helix and somehow make a copy of the A's, C's, T's, and G's in a particular region. What it looks like, roughly speaking, is you have to unzip the DNA. It's a double helix. Like A's, T's, and G's are the ladder steps. You have to unzip that, and then you have to have A's, C's, T's, and G's available floating around that are brought in one by one and stuck next to their complements. So if I have the BRCA1 gene that's located in one particular place in this double helix, if the body can somehow go to that location, unzip, and start plugging in A's, C's, T's, and G's where they're needed, and then detach and just take that new thing they made, you have a copy of the BRCA1 gene. Okay, so that process, one of the things that happens is that a protein might come and bind to the DNA by the BRCA1 gene and kind of sound the call. This is where we need to be. Unzip here and bring in all the A's, C's, T's, and G's to make a copy. A transcription factor is a protein that binds to the DNA and serves to regulate the expression of a nearby gene. So we need BRCA1 protein for some reason right now. There might be a transcription factor that's a specific protein that gets a signal to go to the right place of the DNA, bind, and by it binding, it initiates this process. Mike is one of those types of genes. There's lots of these transcription factors. We're talking bioinformatics, and roughly speaking, I've said, for our purposes, statistically thinking, our data are often going to look like an M by N matrix of numbers. M features, N samples. What goes in there, what those actual numbers represent, will vary a lot from application to application. But roughly speaking, we're quantifying, how many copies of something there is, how active a particular gene is, that kind of thing. There are technologies that are specifically for assaying, getting data, quantitative data on transcription factors. We'll mention that just briefly, but just as an aside, bioinformatics, think of an M by N matrix of numbers. What the features are, what's represented in the rows, will vary from application to application. There are applications where the rows are transcription factors. All right, so Mike is a transcription factor, and it's an important one. You can read the article. I'll let you look at it on your own, but it's involved in, it has a known role in the process of cancer. In particular, if your sequence of Mike gets mutated, you mutate the ACTG sequence, maybe you insert a base, or you delete a base, or more than one base, or you just change an A to a G or something. Happens through just random things happening, happens from DNA damage due to exposure to different things. And if this gene gets mutated, then it's not going to operate like it's supposed to. And in particular, when this gene is mutated, it causes that transcription factor to always be on. And apparently you don't want it to always be on. If a transcription factor is on, then its corresponding gene, whichever gene it regulates, or genes it regulates, are going to be regulated. So in this case, if the mic gene is on, coding for this transcription factor, then that protein, copies of that protein are always floating around, which means that copies of the genes that it regulates are always being turned on. And that can cause cancer, apparently, or that's associated with cancer. All right. So let's come back here, and I think we already ran. Let's get the sequence for that particular gene. Another use of the Biomart functionality. I have a specific gene. Let's go get, in this case, the amino acid sequence of the mic protein. I'm going to say seek get sequence. ID is mic. That's the HGNC symbol. I have to specify that I am giving you an HGNC symbol, as opposed to, say, an entree code or an ensemble code. Different codes for the same thing. I'm using an HGNC symbol, so I had to specify that. I specify that I want the sequence for the peptides, which will be amino acid letters, and I have to give it the connection to Biomart that we created. When I do that, I can use the show function, which is a built-in Biomart function for accessing an object of this class to see the actual sequence. Okay. Used with the ensemble Biomart. Can you guys run that? Works for you? Yes. Okay. So the type, we do question mark get sequence. Question mark function name always brings up a page like this. And here in the argument section, it should tell me some detail about what each argument is for. The type argument needs to be one of these. So these are what naming convention are you using to indicate the gene you want? We've talked last time about these HGNC symbols, like BRCA1 is an HGNC symbol. Ensemble has its own code for that same gene. We'll see another database called Entree. It has its own code for that gene. So whenever we said ID equals MYC, we were referring to an HGNC symbol, and then I needed to tell it that when I gave it the type argument. The other argument is seek type, and that specifies what kind of sequence do you want. So do you want A's, C's, T's, and G's, which tells you the DNA sequence. It refers to it as cDNA, as in complementary DNA. For our purposes, it's just A's, C's, T's, and G's. It could be peptide, which is amino acids, and we won't mess with the other three. So this runs on my computer. Sounds like it runs on your computer. When you type show seek, you should get letters. You get letters on the screen, not A's, C's, T's, and G's, but L's and S's and such. Yes? So if you have, let's say you have a protein and you have a symbol for a protein, how do you know like what type of symbol it is? There is a finite number of conventions. So I guess I would have to search in the metadata for whatever data set I have. Surely somewhere it's going to say in the fine print or in the comments section. You could Google the ID, like the actual ID, and sometimes get what you want. Like if you Google Mike, it'll quickly get you. You can figure out it's a HGNC symbol. There's other types of IDs. So it would, I guess it would just generally would take a little bit of detective work to figure out what code am I looking at. I might Google protein naming conventions, common protein naming conventions, and look at examples for the first 3 most widely used. Does mine look like that, yes or no? That's what I would do. Okay, when you do this, you get multiple sequences, right? The reason that you see each sequence, each little character string of letters is an amino acid sequence that's known for this particular gene. We talked last time about the fact that any one gene can actually turn into multiple protein products, depending on what the body needs at the moment. So the Mike gene, when you print show seq, you are getting from Biomart, these are all the known protein products of the Mike gene. And they all might do slightly different things. So then you could look those up on the HGNC website and learn more about each of them individually. So the point so far is just that Bioconductor is a big resource of our code for doing bioinformatics stuff. One package is Biomart. Biomart's handy for getting sequence data. You have a specific gene, you have a specific protein, and you want to find the sequence, and you want to look it up and learn more about that gene. That's a common task that you would want to do. Let's keep moving. Another issue is that when you're doing this type of laboratory technique, you get a tissue sample from a mouse, and you're going to go get bioinformatic data off of it. As we'll talk about, there's different types of technologies that can be used, and they can be used to answer different types of questions. One type of question that you can ask in a bioinformatic way is, if you think back to that central dogma idea, you have DNA, DNA is copied into RNA, RNA is then translated into protein. So if you want to know at a molecular level what's going on in an organism, you could query the DNA. You could sequence the DNA. What is the sequence of this particular specimen? And look for differences, say, between healthy and unhealthy. Are there mutations? Are there deletions? Things like that. That's just the reference manual, though, like the hard-coded information about that organism. You could also try to quantify how active the genes and other places in the DNA are. The Mike gene, is it on right now or is it off in this tissue sample? Is it relative to other individuals of this species? Is it up or down? So you'd like to be able to quantify just how active the gene is. And if you think about DNA to RNA to protein, if the Mike gene is turned on, then there will have to be copies of it, RNA copies of it floating around. That's the only way that you get the protein. So if you want a lot of the protein, you need a lot of copies of RNA. So you could try to count, roughly speaking, count or somehow quantify how many representatives of the Mike gene are floating around. And one of the earliest technologies that's kind of chalked up as bioinformatic, generating data is called a microarray. So a gene expression microarray. Expression, when we say expression, we just mean, roughly speaking, the activity level of a gene. Is it highly active? Is it not active? And these gene expression microarrays are technologies that are designed to do that. So let me show you a picture. So this on the left side is what's called an Affymetrix gene chip gene expression microarray. Affymetrix is a company, and there's multiple companies that you can buy something like this from. But that's what it physically looks like. So you have 30 mice that you want to do gene expression analysis of, you would buy 30 of these things. And they're each about that big, roughly speaking. In the middle is where the action takes place. And if you zoom in, so we have 30 mice. Each mouse, we extract tissue sample. From the tissue sample, we extract genetic material like RNA. And roughly speaking, we wash that mouse's RNA, all of it. You have all these different copies of RNA. So you've got tens of thousands of genes, each of which can be coding for protein. So if you were to take a sample of all the genetic material in this mouse's tissue sample and use a laboratory technique to pull out just RNA. So there's other things floating around, lipids and things like that. We don't want that, but there's techniques where we can just pull out the RNA. So now I have all the RNA for that mouse's tissue. And on this chip, if you zoom in, so here is looking close at the innards there, and then here is looking even closer at one particular region. The chip is an array with thousands and thousands of little cells, not like cellular organism cells, but just like think of a matrix. It's like a little matrix, and in each of those wells or cells, there are lots of copies. So if you pick one little spot on this picture over here, like this red one right there, if we were to look really close at that, there would be lots of little copies of the same sequence that's complementary to a specific RNA. So suppose we did this for humans. This is a human, specific to humans. On this array would be wells, and each well is specific to one gene. Or there might be replicate wells for the same gene, depending on the design of the analysis. But roughly speaking, each well is a different gene. And at that location are lots of little complementary pieces to the target gene. So like here, let's pretend that's Mike right there, MYC. Zoom in on that spot, and you would see we have our it's popping up this little inset, which is okay, I can work with it, but I don't know why it's doing that. Every time you write, it creates one of these little things. May not be let's just do this next time. It's okay, I can get by without it. That's fine. I'll just do without it. If you were to zoom in on that one spot here, you would see, think of it like a carpet. Lots of little strands that are attached physically to the chip. And so here's my solution full of all the RNA for this mouse's tissue sample. We wash it on top of this surface. And so the mic RNA that came from the mic gene, has ACs, Ts, and Gs, and they're complementary to the spots, the little strands, the carpet there, and only there. All these other spots are complementary to other sequences. So ideally, if you let it wash on there and you let it mingle for long enough, all the mic RNA will mingle over there and only stick at that spot. They will bind to the carpet at that spot. And BRCA1 RNA is going to come over here and bind to its spot. And here's another gene, and there's another gene, and so on. Okay, and then the other kind of magic piece is that when we extracted the RNA from the mouse tissue sample, in the laboratory, you're able to add a fluorescent dye to the sequence. So you have ACs, Ts, and Gs, and then at the end of that, just chemically, you put a molecule that if you shine a laser at it, will emit a light. And what that allows you to do is, after washing all of that RNA onto this chip, you shoot a laser at the chip, and that turns on all the fluorescent dyes. So if at the mic well, it turned out that mic was highly, quote, expressed, there were lots of copies of the mic RNA floating around, there's going to be lots of light because there's lots of copies of that RNA. So we have a bright red, high intensity spot for that gene. Here's another gene that had virtually no representatives in this particular sample. I think it is, it depends on, you may be right, I'm not sure. I thought that it was red as a higher intensity, going from black to red, or black to yellow. I'm not sure what their coloring scheme is. It'll be clear when we get to actual numbers. When we go to an Excel spreadsheet for a bioinformatics data set, M by P, M is all the features, so there'd be one row for the mic gene, one row for the BRCA1 gene, and so on. And then in one column, we would have all the numbers for this one chip. So suppose we had 30 human beings, 15 have diabetes, 15 don't. Each person, so I walk in the door, I don't have diabetes, you take a sample from me, you run it on a chip like this, you have this image for me. You would have this another image for the next person and the next person and the next person. And so when we get to the data, we will just have numbers. And big numbers mean higher activity, more copies. The numbers don't actually equal copies. It's not so precise that we physically get to count the number of RNA there, but it's this fluorescence thing. If there's more copies, you get a brighter light is the expectation. When we get to our M by N matrix, it'll be a bunch of numbers. There'll be big numbers sometimes, like tens of thousands, and all we can really say about them is that they quantify, not perfectly, but they quantify relatively expression level or activity level. So just to reiterate, these types of data are highly multivariate. Multivariate means for each individual in your sample, you get like a vector. You get more than one observation. Instead of just blood pressure for everybody in the room, for everybody in the room, we get 20,000 numbers. Highly multivariate. So my M by N matrix has thousands of rows, one each for every, quote, feature. The feature in this case is RNA, or we might just roughly say genes, because each RNA is a representative of a gene. Features could be other things, like proteins or RNA products and other things, but M by N generally. Okay, so going back to the R code, we are about to, we're going to pretend that we did an experiment with this technology. So we had 30 human beings. Each person has one of these arrays for him or her. And one of the challenges, just a practical challenge, is that the output from this experiment will be numbers. After they've been processed some, you'll have a number for each well. Unfortunately, just practical issue, you're not going to have, for example, a row named MYC and then the number for that well. Affymetrix, it's a company, proprietary company, and they have their own code for the wells. You can find out if you say, here's a well, and it was very bright. It looks interesting because it was bright for the diabetics. It was dark for the controls. So I want to know what gene that is. Because eventually we're going to have our M by N matrix, and we're going to go, certainly one thing we're going to do is go gene by gene or feature by feature and do, say, a t-test. Are the numbers, is the brightness for the diabetics different than the brightness for the healthy people? Do that for thousands of genes, and let's find the subset of them that look different, and then go learn about those. Yes? So I'm not a biologist or a geneticist, and for our purposes, I'm going to say gene, sometimes to indicate just a particular sequence of the DNA. I've explained genes to our audience, which is about half Masters of Statistics folks who don't know much about genetics. I've said, think of a gene in large part as something that codes for a protein. But there are locations in the DNA, sequences of DNA, that are known to do other things besides code for protein. And these spots could be any sequence that you want. As long as Affymetrix knows what sequence to print as the carpet on a well, we can target anything. Roughly speaking, I will say gene to indicate a feature. All right. So let's come over here and see what we can do with Biomart to communicate and learn more about an Affymetrix experiment. Affymetrix human genome microarrays, or just Affymetrix microarrays, because you can buy these things for mice and for a whole bunch of different organisms. If you're dealing with an Affymetrix product, you need to be able to translate its code into like H, G, and C symbols. So I am going to install another package, and this package is also from Bioconductor. It's called HGU133A. That HGU133 refers to this. It's the Affymetrix product called the Human Genome U133 microarray. So there is, go ahead. Say, what? I'm not certain. I think this is specific for this specific array that you buy from Affymetrix. It is? Okay. So there would be different okay. The newest one is +2 or something, right? Okay. So take this, we're going to cover a lot of things throughout the class. This is an illustration of something you can do with Bioconductor and Biomart. Certainly not the only thing you can do is this particular microarray type, but if you find yourself with bioinformatic data that comes from a particular technology and you want to translate its features into features that are more recognizable, like HG and C symbols, you can do it with this code. You might have to change some of the specifics, but in particular, if you went and looked at the reference manual for Biomart, look at the textbook, they have some more examples. So let me just show you how to use this particular one. I'm going to install this package. To do that, I'm going to source in this function that we've used before. And now I'm going to use this installer. So it's downloading something kind of big because we have annotation information for the whole human genome. Might take a moment. Yes? In theory, you can. So the question was, I'll just repeat for the mic. Suppose you have data that doesn't have, say, a package like HG133 specific to it, but you have your own, maybe, genomic information in a separate file. The short answer is yes, you can make it so that you can use these functions to deal with your custom data. It might take some effort to get, because when I was talking about classes of objects, show the sequence that we asked for from Biomart, that object that was called seek was of a specific class, and there's going to be some constructor function from the package that you would have to use to create an object of that class. So the documentation would be in there. My experience from trying to do that is that it can be a challenge. But I mean, if you're writing a PhD and you've got a custom, bacterial community genome that you are working with and you want to use these functions, it can be done. Maybe take some effort. We're almost out of time, and I wanted to, before we quit, make at least a comment about the homework. The homework is due on Friday. The first part should be straightforward to do by just following what's in that R_bioconductor script that we're playing with. So get a connection to Biomart, to the Ensemble database. We did that. And go get the DNA and the amino acid sequences for a particular gene. So I showed you how to do that for the Mike gene, for the BRCA1 gene. You just change it. An ID conversion. So this is an HG and C symbol. Translate that into an Entrez ID symbol. Entrez is a different database. ID conversions enables you to go between, for example, something called an Entrez gene ID and a HG and C symbol. So here's code that shows you how to do that. Right. You can do it, however, if you can find other ways to do it, you don't have to follow my code exactly. The second problem, go look at the Wikipedia article for BRCA1 to answer the first one. I'm not expecting anything more than 2 or 3 sentences. Part B, we're just now doing that. So it's asking about, suppose you want to find BRCA1 on this microarray, the Affymetrix HG133 gene chip. How do you do that? We are doing that right now. So you can use the code here to do that. The only thing that I think we'll have to talk a little bit about is the last one. And I'll say for now, just have a look at the code that you have in this script. Down further, we'll get to it on Wednesday. Down towards the end, I have example code that's specific to that problem. So it's called enrichment, gene ontology enrichment. And again, it's just a matter, in this case, of mimicking the code that you have here. There is a little bit of detail that has to be done kind of manually. So I'm on the discussion boards, holler on there and I'll help, and I'll talk about it a little bit more in class on Wednesday. You can figure this out right now by following my code and maybe looking at the documentation a little bit. But if you get stuck, holler on the discussion board, and I'll talk about it on Wednesday. So let me see where we are back here. So what was the answer? Did we want to say yes or no? Yeah? Okay. I'll just run these 3 lines and then we'll be done. What we're going to do is inside this package that we just downloaded, the HGU133A database, there are several objects that map between Affymetrix features, which it calls probe sets. Basically, that's the carpet that's specific to one gene for our purposes. There's different objects that allow you to map that to other types of identifiers. And one of them is this guy, which maps between the Affy ID and an Entree ID. So I'm creating an object that is that. There's a function called mapped keys, which connects the dots. And then I'm going to ask for show me the Entree codes for the Affy features. Hopefully this is done. It takes a while. You're free to go if you want to. I'm just waiting for this to finish. All this is going to do is tell me codes for each of the probe sets. So the first probe set, first well in here, like this right here. That well, according to Affymetrix, is given the name 1053_AT. And we're going to figure out that that corresponds to the gene with Entree ID number 5982. We could go look that up at the Entree website and learn what it is. We could also, using what we did above, translate between Entree and, say, HG and C symbol. And it turns out that the first well on this particular microarray corresponds to the gene with HG and C symbol RFC2. Do that for your whole Affymetrix data set, and at the end of the day, you might have p-values for each of thousands of HG and C symbols. chances are your biology collaborator would know H, G, and C symbols. If you said, I'm talking about gene RFC2 or BRCA1, chances are they'll know what that means, as opposed to 1053_AT. They probably won't know what that means. So translating between these different naming conventions is kind of a necessary evil. All right, well, I'm done waiting on this, so we'll pick up on Wednesday. Any questions right now then?